Hear me?
Yeah, fine.
Okay, fine. Good afternoon, everybody. So, welcome to the program, first of all.
Yeah, thank you.
And I welcome all the participants to the program. And today's session on application of the push pull technical strategies in the pest management is going to be engaged by Dr. Venkateshalu, sir. And sir has been the professor of agricultural entomology and head of the department, head of the department of entomology in the University of Horticultural Sciences, Bagalkot, and he is university head. And he has an illustrious career of serving as an entomologist. And he has served for 9 years in the University of Agricultural Sciences, Harvard, in the cadre of assistant professor of entomology, and 7 years in the cadre of associate professor of entomology in US, Raichur. And right now, sir is working as the professor and head of the department of entomology in our university, the University of Horticultural Sciences, Bagalkot. And sir has the awards and recognitions credit, and first and foremost is the Indian Council of Agriculture Research Junior Research Fellowship for his MSc studies, and sir has been the recipient of the UAS, the Harvard Gold Medal for completing the PhD degree program in ranking first. And sir has guided the students as a chairman, and he has been a member for the 10 students in the post-graduation studies. And a number of students on roll right now are 12 in number, and sir has handled several externally funded projects, and out of which a few of them are like the evaluation of the watershed projects in Ballari district, which was funded by the Jilla Ballari, and dissemination of insect pest resistance management strategies in cotton in district, that is Gulbarga, and eco-friendly pest management measures against pests in PT cotton from the 2009 to 2011. This was funded by UASR Demand-driven project, and the baseline study of flora, fauna, and proposed uranium mining site at Kogi, Gulbarga district, Karnataka, during the year 2009 to 12. And this was funded by Bark, Mumbai, and establishment of a plant health clinic at the Agriculture College, Bindangudi, during the year 2012 to 13. And this was funded by Indian Council of Agriculture Research, New Delhi. And evaluation of HGW86 10% OD is highest peer against the best of the cabbage project for the year 2009 to 10, and this was funded by DuPont India Private Limited. And evaluation of bioefficacy of PII 504 20% SG on okra against second pest complex aphids, thrips, chassis, and whiteflies. And this was funded by Pesticide Industries Limited, Gurgaon, Haryana. And the next project is current status of bolome complex on PT cotton hybrids, and this was funded by UASR demand-driven project, and establishing the Center of Vegetable Research and Development. This was funded by RKEY. Management of the major diseases and the pests of chili, tomato, brinjal, onion, and grapes using plant growth regulators, and this was funded by Willow Wood Chemicals Private Limited, New Delhi. And evaluation of bioefficacy of thiamethoxam after 25% SG. So as a soil application against the pest complex in grapevine, this was funded by Syngenta India Private Limited. Evaluation of new molecule PII 8007 20% SC on pomegranate, and establishment of semiochemical laboratory. So this was funded by RKEY. And sir has forwarded, and he has developed 5 different technologies as a principal investigator, and he has been a collaborator for the 9. Sir has been a collaborator for the 9 technologies that were forwarded, and research publications. Sir has 30 in number, so which were published in the referred journals, and the short notes in the referred journals, they are 9 in number. The full-length papers in symposia and workshop of repute, they are 8 in number, and the research note abstract in symposia, seminar, workshop, they are 10 in number, and books and extension booklets. So sir has published 3 in number, and Venkateshalu sir has published 30 folders, extension which are extension related, and the selected popular articles are 20 in number, and to mention few of them, like the first and the foremost is the phonistic studies on the genus Xanthoids, so associated with the Bendy ecosystem in Karnataka. So this was published in the Journal of Entomology and the Geology Studies, and diversity of the noctid moths associated with the major vegetable crops in Karnataka. This was published in the Journal of Entomological Research, and diversity of the noctid parasitoids in vegetable ecosystem of Karnataka, and this was published in the Journal of Entomology and the Geology Studies. taxonomic description of the genus Spodoptera from Karnataka, and this was published in the Journal of Entomology and the Geology Studies, and taxonomic description of 3 species of Heliodine, and this was published in the International Journal of Advanced Research. So this is the brief profile. In fact, it has been very large, and the sir has cut it short. So that's why. So this is the brief biodata, the profile of Dr. Venkateshalu sir. Sir, welcome to the program, and you can take the charge for the further proceedings of the session, sir. Please, sir.
Thank you, Dr. Satyanarayan, for introducing, and good afternoon to all participants. Today, I will be discussing on the applications of push-pull strategies in pest management. So, where in the today's very short topic, so this is so in that I will be discussing on the issues like the principles and components of the push-pull strategies, and I will try to explain a few classical push-pull strategies. So in that there is one which is the in the land to lab mode, and another one so which is the reverse of our usual type. So another one is a very recent one which is on fall armyworm, that is the land to lab mode. And so I will also explain about some of the push-pull strategy attempts that are made in India. And so we have our own effort made for the management of fruit sucking moths on pomegranate, so by one of our PhD student. And so with that, then I will finish up my topic by giving some of the future strategies that can be taken up on these aspects. So now to begin the push-pull strategy. So the concept of push-pull strategy was first perceived. The idea was conceived by the Pike and his colleagues in 1987. So when they were working for the management of Venkateshalu species on cotton in Australia. So during that, so they were they conceived the idea, and later on in 1990, that's Miller and Covens. So they formalized and refined, and they termed the technology as a stimulo-deterrent diversion. So these they formulated, they revised the term of push-pull strategy when they were planning for the management of onion maggot with this the same technology. So, however, in both the cases, if you see the original terminology and the stimulo-deterrent diversion, so they appear very similar. So they imply same meaning. So because of that, so in most of the subsequent publications, so the original terminology, that is push-pull strategy, has been retained, and because they imply the same meaning of the stimulo-deterrent, so the push-pull strategy is appeared in most of the subsequent publications. So this is the principle of push-pull strategy, how it functions. So there are basically two components, like one push and another pull component. So it is nothing but a behavioral manipulation of insect pests via the integration of two stimuli, that is stimuli to push from the main crop, and another one the stimuli to pull from the main crop to the source. So here the source will be our predetermined source, and subsequently from that source the insects will be removed. So here in these principles, so the main the thing is that we are employing behavior modifying stimuli in order to modify in order to divert the insects towards a predetermined site, and then subsequently they will be removed. So where so the stimuli will be applied in this the strategy. So the components. There are mainly two principal components in this the strategy, where that is a push component. So in the push component, in the very simple terms, it is the repelling or deterring the pest from the source. So where we will be deterring the pest from our main crop, that is the principal crop. And so we can employ several kinds of push components. So they may be either visual cues or maybe chemical cues. So among the different push components, the visual distractors, which are the visual cues. So we can apply different shapes or colors in this component. And the another component is the non-host volatiles. So these non-host volatiles mainly they work like a repellents. and important the areas where important the components that we can explore here are plant essential oils, which work like the repellents. And there are also the waste derived semiochemicals. So waste also gives the cue for the insects to attract. So such waste derived cues mainly the herbivore induced plant volatiles. So which are released when there is a damage by the herbivore. So such of the waste the derived semiochemicals can also be used in the push component. Then anti-aggregation pheromones, which is so this is a multiple mode pheromones where at lower concentrations they act like the attractants, at higher concentrations they repel. So because of that they are known as anti-aggregation pheromones. So they mainly act like anti-aggregation at higher the concentration. Then alarm pheromones, so they are mainly employed, or they are mainly used by aphid group of pests, and we can use those alarm pheromones also. And the anti-feedant and ovipation deterrent and ovipation deterrent and ovipation deterring pheromones. So these the anti-feedant and ovipation deterrents are from the again from non-host the plants only, and we can also use ovipation deterring pheromones. So these are released mostly by the females to mark the explored areas. So they are usually employed by the female insects to mark the explored areas. After ovipation, many of the dipterans utilize these ovipation deterring pheromones. So all these components can be used like push components to push the pest from the main crop. So then another component is a pull component. So we will be pulling on the other end. So where the stimuli are employed to attract the pest towards our the predetermined or targeted source. So this source will be in the companion of the main crop. So here the pull components includes visual stimulants and trap crops. So the trap crops largely are involved in the pull component. So various trap crops can be applied, and host volatiles can also be used. So where host induced plant volatiles works in both the ways. So they can work like the attractants as well as repellents. So they can be used in both the stimuli. And the pheromones, so where the host finding is usually applied by these pheromone pheromones. So then the aggregation of pheromones, where the lot of aggregation pheromones are used by coleopteran insects, and sex pheromones largely employed by lepidopterans. And so we can also use ovipation stimulant, gestated stimulants. So again, these are produced by non-host the host plants, so which for attraction towards the ovipation or feeding. So these are the pull components that we can employ in the push-pull strategy. So these are the two the principal components in this. So apart from these two principal components, so there is one more hidden component in this strategy. So in most of the cases, it is not highlighted, but it is the most important component in this. That is the hidden component is the population reduction methods. So that means usually in the push components, we will push the pest from the main source towards the targeted or predetermined site. That is towards the predetermined source using pull components. Once they are accumulated in the pull component or pulled area, so then there is a very important thing to reduce the population in that area. Otherwise, there is a likely chance of further multiplication in the predetermined area, and they can shift back to the main crop. So that's why it is very important to eliminate the pest from the pulled sources or from the predetermined sources. So there are various the population reduction methods can be applied. So one is the dead end trap crops. So these dead end trap crops are nothing but the plants which are highly attractive to pest, but their survival will be very less. So that means the insect which are attracted for laying the eggs, so the larva may not survive. So because of that, the population naturally comes down on the trap crops. So such of the crops, trap crops, we call them as dead end trap crops. They manage on their own. Then suppose if the trap crop is not of a dead end kind, so we can employ other techniques like biological control. We can explore the usage of predators, parasitoids, and so especially the conservation. So where we can plant the pollen yielding plants. So most of the predators and parasitoids are dependent on the pollen. So we can conserve them by providing pollen yielding plants. And the augmentative religions can also be made. So where it is possible to implement in a smaller area. So augmentation is required in a smaller area rather than in a bigger area in the main crop. Then we can also use the host-induced plant herbivore-induced plant volatiles. So where these herbivore-induced plant volatiles also gives the cues for predators and parasitoids to get attract towards the host. And so such apart from this, even we can use the food lures and supplements also to improve the population of biological control agents. Then biological insecticides can also be used like the BT, entomopathogenic nematode, entomopathogenic fungi, and NPV can also be used on the full component to eliminate the population. Then suppose if it is a active stage, so we can even use the mass trapping devices. So various mass trapping devices can also be applied at the full component. Suppose if all these doesn't work, then lastly we have to go for the toxicants like chemicals or botanical insecticides. But originally when the concept was devised or concept was conceived, so it was thought that the entire system should be non-toxic. So it should be as far as possible non-toxic, but later on, so various modifications occurred, and because of that, so even the toxicant application was also included. So the main the concept or main logic is to reduce the usage of toxicants. So here, so this is the principle how the push-pull strategy works, where initially we have to push from the main crop, then we have to pull at one end, then we have to manage the attracted population using various methods. So why we have to use the push and push-pull strategy? So in fact, so these the individual components of the push or pull components, they are also they can also work on their own or individually, but their individual component efficacy is much less compared to the broad spectrum insecticides. So they cannot be comparable with the effect or efficacy of the insecticides. So in that sense, it is very important to integrate these push and pull components and deploy them in tandem, so one after another, so that the efficacy of the components like the push pull components can be additive. So individually they will be effective to the extent of 30 to 40%. So the additive effect will be much more than the 80 or 90%. So that it is possible to get higher efficacy by deploying them in tandem. Then it is another important thing. So it is the best way to explore ecosystem services. So like in the nature, there are many the biological control agents will be acting to reduce the population. So many times the population comes down on their own. So such of the ecosystem service we ignore in most of our IPM modules. So this is a very important strategy where we can explore the ecosystem services for the benefit of the pest management. So with this, the idea, the push-pull strategy is developed. So here it is important to integrate these components to develop like a holistic the push-pull strategy. So some of the important the classical efforts that are made to develop the push-pull strategy are done in sub-Saharan Africa, where for the control of the stem borers in maize and sorghum. So this is a great successful push-pull strategy that worked in Africa. So where if you take the maize and sorghum, they are the principal crops for the poorest people in eastern and southern Africa. And so these crops are largely damaged by lepidopteran stem borers like Tylopartella, Sesamia, and Eldana and other species of stem borers. So they are a complex of stem borers. So they account nearly to the tune of 10 to 50% yield loss. So this is a huge loss for the poorest people. So with that, keeping that thing in their mind, so they developed some of the pushable components. So where they explored the push possible crop plants or the companion plants like the molasses grass, silver leaf desmodium, and green leaf desmodium. So these are the crops that were used in the push component. And in the pull component, so usually the forage grasses were used like Napier grass. So this Napier grass is excellent the component to attract the stem borers. So they used the Napier grass in the pull components. So they evaluated these systems. So that means the push-pull strategies in Kenya. So where during they evaluated this system in 1998-99. So that was for the first time it was evaluated in a systematic manner. So they taken the studies in 2 locations in 2 years, like in Trans Nigeria and Suba in Kenya. So they adopted 2 systems of the cropping. One is maize plus Napier. So the maize is the main crop and Napier is the pull component. And then another another module, they have adopted maize plus Napier plus desmodium. So here they have included desmodium as a push component. So Napier is a pull and the desmodium is a push component. So in the maize they adopted. So in the suppose if you take this the maize, Napier and desmodium, so the desmodium is planted as an intercrop in between the maize. So whereas the Napier is planted on the periphery as a pull component, which is attractive. So this the 2 models they evaluated for 2 years, and it has shown that the stem borer damage in the first the mode, that is only in the pull component. So it was not much. The stem borer damage was not reduced much. So whereas if you take the push and pull together, so the stem borer damage has reduced significantly compared to the sole maize crop. So it was almost less than 10% in most of the areas and years. So also because of this, so they adopted the maize, Napier and desmodium cropping system. So that means desmodium is used as the push component and as an intercrop to plant between the maize rows, and the Napier is used as a pull component, and they planted that one in the periphery of the maize field. So here the Napier acts as a dead end plant. So this the Napier grass, that means the larvae of the stem borers that are feeding. So usually the survival rate will be very, very low. So though the moth deposits heavily on the Napier grass, so the larva cannot survive because the plant produces gummy substance. So because of that, the larval survival will be reduced, and because of that, the Napier grass acts as a dead end plant for the stem borer. So further, they looking at because of the success, so they taken up the further studies in another 14 districts of the Kenya. Subsequently, where they adopted the push-pull system, that is the maize, the desmodium and Napier system, along they compared with the maize monocrop in all the 14 locations. So the grain yield of the maize was significantly high. So it was much more higher than the mono maize crop system. And so this where they planted the desmodium. You can see the photos where the desmodium is the legume crop. It is grown as intercrop between the rows of maize, and the Napier on the periphery. And so most of the farmers, they accepted. Socially, it was quite acceptable because of the higher yields. And most of the farmers were attracted towards this technology. And this technology has been adopted to a larger extent. So in the sub-Saharan Africa, in fact, so initially it was tested in 2 districts in Africa. Subsequently, it has been so we are disseminated to many other districts in sub-Saharan Africa. And so as per the 2014 data by Jayur Khan, so these the person responsible for developing a strategy and disseminating it in Africa. So where they the year-wise the adoption of the farmers increased gradually, and so where in 2013 almost 68,689 farmers adopted this technology. So if you look at the adoption level, it shows how the sound is the technology. So the adoption level is very high. So and most of the farmers adopted very easily this concept, and the concept was very simple, and so it was easy for them to grasp, and they adopted it very easily. So apart from this, the efficacy in maintaining or managing the stem borers, so this system, the push pull system, has additional benefits. So like the desmodium, if you take the desmodium, apart from repelling the stem borers, it also suppressed the striga, so which is a the weed in the maize. So the suppression of striga was very significant, and so because this is a pulse crop, so it has improved the soil fertility. And with respect to the Napier grass, so it is the forage crop, so they used it as a cattle feed. So it shows that, so whenever we select the trap crops or companion crops, the companion crops must have a multiple benefits. So where they must be of the harvestable type, not the sacrificeable type. So we should harvest them for our better, for improving yield or for other purposes also. So because of these additional benefits and because of the efficacy in managing the stem borers, so this technology has been disseminated in larger area. And so this later on they started to understand how this push pull system works. So initially they adopted this one in the field, then they brought it into the lab. So this is why it is a kind of land to lab mode, so rather than lab to land mode. So where they identified that the regions for the pushing the stem borer from the main crop or the maize is because the desmodium produced 6 volatiles which are repellent in nature. So these 6 volatiles are responsible for pushing repelling the moths from the maize. And so they also identified the 4 components in the Napier which are responsible for attracting the stem borers. So it is very important to attract the stem borers. Otherwise, so our push strategy may not be a perfect the type. Then they also found that, so this desmodium, wherever the desmodium grows, so this desmodium root exudates suppress the striga. So when they analyzed it chemically, so they found 4 the allelopathic chemicals which are responsible for suppressing the striga. And so because of these additional benefits are where it is a semi-chemical based approach, so where the insect behavior is modified. And so this is a classical example of how a push-pull strategy, if it is feasible and socially acceptable, so probably it can spread on its own to a greater extent. Then there is one more the recent effort made for the management of push-pull strategy for the management of the Spodoptera frugivorida, that is the fall armyworm. So this effort is made in Mexico. So it is a case of the lab to land. So where the components are evaluated initially in the lab, then they will be promoted to the land. So the potential they identified the potential attractants and repellent plants which are suitable for designing a push-pull strategy. So they identified 6 attractant crops which are basically of the forage crops. Then they also identified non-host the repellent crops. So which includes like the Tegetus erecta, that is the marigold also. So they evaluated these 6 attractants and repellent the crops as a the push and pull the pull and push components respectively through the laboratory studies. So where they conducted series of the studies to know the oviposition response of the Spodoptera frugivorida on various attractants. So they found the difference, so where significant difference among the host plants with respect to oviposition. And so they also conducted two choice tests. So in the 2 choice tests, So there are few the plants like the Mombasa, so that is the Penicillium maximum Mombasa. So this showed significantly higher number of the eggs were deposited on this grass compared to the maize. So such of the crops were evaluated in a two-choice test. And further, they have taken up this in a 4-arm olfactometer studies. They also conducted 4-arm olfactometer studies in order to strengthen the attractive the modes of the attractant plants. So in that case, where they have conducted the behavioral response attraction studies, so which the attraction studies includes there 3 scenarios. They have, so this is the depiction of the 4-arm olfactometer. So where there will be 4 arms. So in each of the arms, we can place one the stimuli. So either repellent or attractant stimuli we can place. So these 3 scenarios represent the attraction studies. We have already understood those in the previous studies. So only I am interested to show you the scenario in the 4th, that is the repulsion. So in the repulsion, they tested the repelling plants along with the maize. So they compared the maize along with the repellent plants, and they placed it in the 4 arms of the olfactometer, and they studied the repellent properties. So among the 4 the plants, repellent plants, so except this, that is except the lucida, Tegetus lucida. So all other plants were proved like a very effective repellent plants. So where except this Tegetus lucida, so other plants were selected like a best repellent plants. And further, they taken up other studies like the feeding preference studies also they conducted. So feeding preference between the maize and the alternate attractant plants also they conducted. In some cases they got very significant variation, the maize and grass, and so they also studied the survival rate. So this survival rate is very important, where the survival rate on some of the attracted plants must be very low. So that they can be a very good dead end trap plants. So among those, so the 3 were recognized as the best plants, like Penicillium penicum, Maximum Tanzania, then Mulanto, and Mombasa. So these were selected for the further studies. So in fact, they developed the model to test to select the best plants. So where they developed trap plant selection index. So considering all the various kinds of the parameters of the insect feeding parameters, oviposition parameters, life cycles, and the survival rates, all considering all those, so they developed the trap selection, the trap plant selection index, that is the traps. So they classified these 6 attractant plants, and they given a rank. So from that rank, so the 3 plants were the ranked. First 3 plants were selected for further studies, like the Mombasa, Tanzania, and Mulatto. So these were evaluated not only for oviposition, even for the survival, life cycle, and several other parameters were also compared. And so those were compared using the traps index. So this is finally after conducting series of experiments, they have developed or they proposed 9 push-pull strategies for the management of the fall armyworm in Mexico. So where they identified 3 push components. So where the push components will be placed in the middle of the main crop, that is the middle, they will be placed as intercrops, and the pull components will be placed at the periphery or at the border. So they at the perimeter of the main crop they will be placed. So they included the 3 pull components. So these the 3 push and 3 pull components. So they finalized based on the traps index, and so now they have to test these 9 push-pull strategies in the field level. So this is how they developed. They assessed the quality or the efficacy of the push and pull components in the laboratory, and they will progress it towards the land or in the field for evaluation. So these are the two successful components, successful strategies that are adopted. So in India, if you take the push-pull strategies, the attempts are very less, and in fact, so they were not attempted like a push-pull strategies, but they were attempted in the context of trap cropping only. Later on, they have been modified to reflect the push-pull attempts or push-pull strategies. So these the push-pull strategy attempts, of course, they have been made in cotton and other ecosystems also. But here I will be concentrating only on the horticultural crops. So there is plenty of opportunities for the scope for adoption of the technology in horticulture crops. So the most important because the horticulture crops are relatively confined, grown in confined areas, so that the operations will be in confined areas, and in most of the cases they will be of high value crops, and so the farmers or consumer wants residue free material where they are consumed fresh. So the residue free material is produce is very essential in the horticulture crops. So because of that, the push-pull strategy has a lot of scope in horticultural ecosystem. So there are a few attempts that are made to develop push-pull strategy or to modify the existing system into push-pull strategy. So one is in the management of root borer and tomato. So this was made, attempt was made at IHR by Srinivasan and his our colleagues. So where they used African marigold as a trap crop, and they conducted series of experiments for about 3 to 4 years. They conducted experiments, and finally they emerged out with the recommendation that the African tall marigold can be used as a trap crop at the row ratio of 16 tomato and 1 row marigold. So this the trap crop finalization was finalized looking into the efficacy of the trap crops in reducing the fruit borer damage. And then another important thing what we have to take care here while planting the tomato. So we should plant the 40 days old marigold along with the 25 days old tomato. So this is to synchronize flowering. So because ultimately, so the fruit borer lays eggs on the flower buds only. And so once it lays eggs and flower buds, subsequently, in fact, so there is no the separate management or the population reduction method recommended. But here, always we should always collect and destroy the larvae which are occurring in the marigold. Otherwise, there is a likely chance of shifting of these larvae to tomato, or they may complete life cycle here and they can shift to the tomato. So that's why. So this the recommendation needs to be slightly modified. Where we don't have specific the push component here. So only the pull component we have adopted, and there is no specific push component. But so it reflects somewhat like a push-pull strategy, but not exactly the push-pull strategy. Then another the case is Indian mustard as a trap crop for the management of the lepidopteran pests in general, and specifically like the DB. So this is also made at the effort is made by Srinivasan and Krishnamurthy at IHR Bangalore. So where they conducted series of experiments to study the preference of egg laying and the preference of larval feeding and all those things. And so based on that, they recommended, so the current recommendation, what we can say in the cabbage is the growing of Indian mustard as a trap crop at 25 rows of cabbage and 2 rows of mustard. So these 2 rows of mustard, we have to sow 1 row 15 days prior and second row 15 days after the cabbage. So this is to maintain the fresh the foliage for the egg laying by the DBM. Then so they have also recommended the population suppression method on the trap crop, like spraying of dichloromas. So the dichloromas has been recommended because it is having very short residual action, and so it is very good the insecticide which affects very quickly. So they recommended dichloromas on the trap crop, and subsequently, in fact, it was modified to add push component to this the recommendation, where in the original recommendations of the Srinivasan and Krishnamurthy it was not there, but subsequently it was added that spraying of NSK 5% or neem and Pongemi oil 1% on the main crop was recommended as a push component. So this kind of the modification has been taken up, but there is no serious efforts to made in India to further made it much more effective or further make the push pull strategy as effective strategy. So these are some of the efforts made in India, and so very recently, in fact, it was also shown that the Indian mustard, though in the original recommendations they included dichlorova spray for suppressing the pests on Indian mustard. So it was shown in the Africa that so the Indian mustard act like a dead end plant. So here you can see they evaluated the occupational preference of DBM on various cruciferous crops like Chinese cabbage, broccoli, cabbage, and cauliflower, and Indian mustard. So they have shown a significant preference for the Indian mustard. But when you come to the larval survival on the Indian mustard, the larval survival Indian mustard is significantly low compared to the cabbage, cauliflower, or broccoli. So it shows that so the Indian mustard acts like a dead end crop also. So whatever the larva that survive, usually they cannot complete the life cycle on the Indian mustard. And very lately, in fact, very recently, the Sarvan Kumar, they have shown that the allelochemical or the semiochemical that is responsible for attraction of DBM towards the Indian mustard, they have identified it as allyl isothiocyanate. So that is the chemical which is the semiochemical which is responsible for attracting the DBM towards Indian mustard. So these two things have been added. So there is a need to revise and revisit and revise the push pull strategies in cabbage as well as in tomato also. So in our institute, we also made attempt to develop management strategies in the context of similar to that of the push pull strategies for the management of fruit sucking moths. So this was carried out by one of our the PhD students, Mudha sir. So as a part of his PhD program, so where so these the fruit sucking moths in horticulture crops, especially the fruit crops, is a very important threat. That to pomegranate, which is a very high value crop, it is a very serious threat. And so this the pomegranate fruit sucking moth is is a exceptional pest where in most of the other lepidopteran pests we see the larva damaging, but here the adult cause damage. So the adult stage causes damage, and these adults are nocturnal. So they come to the pomegranate or even other fruit crops fields also, and they suck the juice from the ripening fruits. So they attack the ripening fruits. So they will be about to harvest and they will be attacked by the fruit sucking moths. So because of that, the farmers feel very unhappy because so the crop will be at the harvest level and they will be damaged by the fruit sucking moth. So that's why so they see this pest as a very serious pest. And in fact, damage symptoms you can see here. The moth, they suck the sap by inserting the proboscis into ripening fruit and suck the juice. Afterwards, the juice oozes out from the hole, and subsequently in few days, the area around the hole will be sunken, and subsequently such fruits will, in most of the cases, they drop out. And so here there is one more important thing that we should remember. So these adult moths, they live for about 20 to 25 days. So in fact, in those 20 to 25 days, they can damage around 20-25 fruits. So the loss of 20-25 fruits by a single moth is a huge loss by the farmers. So because of that, they feel this is a very serious pest, especially at the verge of the harvest. And so when we took up the assignment, and when we visited the field during night hours, it was surprising to see several the moths visiting the ripening fruits. So there are several the moths we noticed, and there are a few the primary fruit sucking moths like the Eudecima materna, Eudecima colonia, and Omena. So there are around 7 species collected as a primary fruit sucking moths, and there are several number of very high diversity of secondary fruit sucking moths we notice. So these secondary fruit sucking moths doesn't cause damage to the fresh fruits. So usually they suck the juice from the split fruits or already damaged fruits. So where we differentiated the primary and secondary fruit sucking moths based on the proboscis. So proboscis tip in the primary fruit sucking moths will be very highly sclerotized, and you will see sclerotized the barbed tip of the proboscis. Whereas in case of secondary fruit sucking moths, the proboscis tip will be smooth. So based on the proboscis, we can easily say whether the moth that is active in the field, whether it is a primary fruit sucking moth or secondary fruit sucking moth. So in fact, as such, the only primary fruit sucking moths cause damage to the fresh intact fruits, not the secondary fruit sucking moths. They usually feed on the fallen fruits or damaged fruits only. So then we initiated to develop some of the push-pull the strategies for this pest. So where we evaluated some of the pushing components initially, and I conducted some of the laboratory studies to evaluate some of the botanical oils. So we wanted to use the botanical oils which are available easily to the farmers as a the pushing components, like they will act like repellents. So in the laboratory studies, it was shown that the combination of neem, lemongrass, and citronella were quite effective in reducing the fruit borer damage and the fruits compared to any other the oils. And so this was further we tested in the field. So where as a push component we tested in the field. So we tested with the various oils like neem oil, pongame oil, NSKE, citronella oil, lemongrass, mau oil, and combination of all the three, that's neem, lemon, and citronella, which proved very good in the laboratory. And azadirachtin, so the sticker alone and untreated. So these treatments were evaluated for fruit sucking moth damage in the 2 years. So across the 2 years, so the combination of all the three proved very effective. So this proved promising both the years, and so because of that we wanted to use that as a push component. We finalized that one as a push component. Then another component we wanted to evaluate like a pull component. So the pull component, so this the fruit sucking moths are they can feed on variety of the fruit crops. So because of that, we wanted to know which the fruits are quite effective in attracting the fruit sucking moths. So he evaluated, so he conducted this study in the net house condition, so which should represent or which should depict the field situation. So he hanged the fruits. So we selected banana fruit, guava fruit, citrus fruit, pomegranate, sapota, tomato, brinjal, apple, and so untreated control as the sponge. So when it was evaluated for several nights, so it was found that the banana fruit was quite effective in attracting more number of the fruit sucking moths. So they land on the banana fruit and they suck the juice. So one important the character is that when they land on the banana fruit, so they can insert the proboscis and they remain on the fruit for about 20 to 25 minutes. So they, unless you disturb, so they continue to feed for about 20 to 25 minutes. After complete feeding, so they will fly off. So finally we found that the banana fruit is excellent for as a pull component to adopt this strategy. Then the other important, once it comes to the banana, means it is important to trap them. So the population suppression device we need to adopt. So because it can land in the banana, but manual collection of the moths from the banana is a tedious job, where they are quite active in the night time. So the farmers cannot spend their time in the night, and it is also impossible to collect all the moths manually. So because of that, we wanted to develop the collection device for the fruit sucking moths. So we evaluated different kinds of traps which are suitable for the fruit sucking moths, and so we evaluated different traps like white cloth trap, black cloth trap, nylon net trap, bucket trap, and bottle trap. So they will be of around 1 meter length, and they will be hanged in the field. And So he recorded the number of the primary fruit sucking moths and secondary fruit sucking moths that are trapped in. So here the attractant used is a banana one. So we wanted to go with the banana. So the banana was used as an attractant. So among the different traps, so nylon net trap has attracted more number of the primary fruit sucking moths. So that was quite interesting. So where it could attract more number of the traps. Once they're trapped inside, so they cannot escape. So we designed the trap in that manner that so once they are inside, so they cannot escape. So only they have to die and they have to fall in at the bottom. So we have to remove them afterwards. So these traps are quite effective in attracting and trapping the moths. And even many secondary moths are also collected in those traps. So we thought it is a very good device to go with the attractant, and we finalized nylon net trap as a good device for attracting the moths. And subsequently, we wanted to develop the modules to incorporate all these components like the push component, pull component, and the collecting device as one, and we evaluated them in the IPM mode. So where we developed, we formulated the IPM modules like attractant module where there is only nylon net trap, then the stimulate deterrent or push-pull the strategy where we adopted the attractant, the nylon net trap along with the attractant source, and the pushing strategy with the essential oils. And in the IPM strategy, we adopted the nylon net trap, then alternate spraying of the repellent and the insecticides. Whereas in chemi-intensive, only the insecticides were sprayed, whereas in untreated control, it is nothing given. So these the 5 modules were evaluated in the field, and we wanted to know how the fruit sucking moth damage can be reduced. So among the different the modules, it is shown that the push-pull strategy, that is push-pull the strategy comprising the pushing component like the repellent and the pulling component like the attractant and trapping. So this has showed very good, and so it is on par with the integrated control. So it is almost on par with the integrated control, both in terms of fruit sucking moth as well as fruit borer. So in that sense, so it has reduced it has recorded very low number of damaged fruits, and so we also recorded the number of moths that are trapped in the nylon net trap. So almost all the nylon net traps in most of the modules, they recorded the huge number of the primary fruit sucking moths as well as secondary fruit sucking moths. So this is the number of moths trapped per week per trap. So the activity of the primary fruit sucking moths will be quite high, especially during the October-November months. So that's why we get a very huge number of the moths per week also. Then we have assessed that the fruit yield in all the 3 modules like attractant, push-pull, and integrated modules are quite good, and they were parallel. So they were on par, where they were very good compared to untreated and of course the chemi-intensive, only chemical module. So finally, we thought to final further. So this has been tested for 1 year, and we wanted to test further. And there is another important thing is that so this the push-pull strategy, which is also when you compare only the attractant module, so this is also which is comparable with the push-pull strategy. So this made us to devise or to make further improvement in our modules. So because of that, we need to test those components. So we developed or we formulated some of the possible the other modules that needs to be tested. So because our existing module shows that even the attractant alone can give. So, but here when the attractant alone is used, the number of traps required will be more. So in order to reduce the number of traps required and in order to make it much more the feasible, so we developed some of the, we formulated some of the modules which needs to be tested. Like so, this is what we place the traps in between the crops in the test module. So we need to test this one as a companion. So where so we would like to place the nylon net traps only in the one area, one part of the small part of the field, and other area we would like to treat with the push component like the repellent oil or some other and the push component. And so the another module, so we need to place the traps in the periphery. So because the most of the moth activity that occurs or when the moth comes from the neighboring fields, so usually it affects the neighboring the plants or the border plants. So it is very important to test this one also, where there is a possibility of reducing the number of traps per hectare. Suppose if you test these modules. So now we are due for testing these modules in the field for further validation of this IPM, this the push-pull strategy in pomegranate. So this is the work what we have done here, and some of the push-pull strategy. In fact, so what the successful case, what we have learned from Africa, so that the successful case is a example for subsistence farming. So that means it was a kind of subsistence farming for maize and the sorghum systems. Whereas if you adopt that same in the intensive agriculture, there are some problems where so many times it may not match with the intensive agriculture. So there is a need to improve the push-pull strategies in order to meet the requirement of the intensive agriculture also. So in that context, so the one very important thing that we need to do in the push-pull strategy is very important to understand the pest biology. So the pest biology gives a lot of idea to decide the push-pull strategies. And also there is a now the behavioral chemical ecology interactions with its host, conspecifics, and natural enemies needs to be understood. So this is another very important aspect where most of the time we should make it non-toxic. As far as possible, we should make the system non-toxic so that the natural enemies can be harnessed for management of the pests. And the integration of habitat diversification with semiochemicals. So semiochemicals plays a very important role in both the push as well as pulling component. So it is very important with the existing analytical techniques. So it is possible to identify the semiochemicals and to formulate them in a suitable way so that we can adapt that in the strategy. And so this system is one possible chance to harness ecosystem services. So in most of the cases, when we go for natural enemy augmentation or conservation, so we consider the entire main crop area as our target. Whereas in push-pull system, so only the pulling area. So the small pulling area can be considered as the area to be managed with the natural enemies. So there is a plenty of scope for exploring the natural enemies only in the confined areas or small areas. And the another thing, it is important to blend with IPM. So there are many other pests. Usually push-pull strategy works for a small group of pests or very confined, very one or two pests. So other pests will not be included in this. So it is important to blend with the IPM for other pests also. And so this the push-pull system is definitely a very good approach to develop in the protected and organic situations. Okay, thank you. Thank you.
Okay.
So thank you, sir. Thank you very much. So now, Dr. Venkateshalu sir has completed the presentation. So if there are any specific queries from the audience side, you can please. If there are any specific questions from the audience side, so you can please answer questions to the Venkateshalu sir.
Hello.
Hello.
Hello. Sir, this is Gangadhara from College of Horticulture, Bengaluru, sir.
Okay, Gangadhara, please.
Yeah. Sir, first of all, thank you for a nice presentation. Sir, few clarifications.
Yeah, please.
Yeah. The first one is, regarding the maize study conducted in Southern Africa, in Kenya. It was a classical study actually,
Yeah.
where they used Desmodium as a push component and Napier as the pull component.
Yes.
Basically repellent, and Desmodium acted as a repellent and Napier as an attractant crop. So where large number of farmers adopted that technology, almost around 69,000, something like that. Sir, after that, is there any efforts where they extracted the repellent, components from the Desmodium, sprayed on the maize crop as a repellent, and the attractant components from the Napier used as the food lure component? Is there any such kind of the efforts made after the getting the successful result from the push pull technology?
Actually, that is correct. But what I have come across, so they have identified the molecules, that is the volatile molecules in both the systems. But subsequently, they have not they explored that. But their theory says that they can be explored in other countries, because in Africa, so it is excellent system where it is working, and the farmers are adopted it. So there is no problem for them. Suppose if it needs to be explored in other countries, probably we can think of using the same the components, or we need to study our own ecosystem. So because the chemical profile may change, even it may change from place to place, but they have not made any effort in Africa to adopt as a spray component. And there is one more thing is to develop the semiochemicals as a spray components. So in fact, there are many other hurdles will come, like the registration. The registration protocols, probably nobody has tried them with respect to the semiochemicals. So because of that, probably there are very few the efforts made, or no effort made to use them in the practical sense.
Yes, sir. Actually, sorry to intervene, sir. Actually, the question which Gangadhar actually asked is like, is there any effort that is made, like, to spray the extract? So not in the context or the perspective of the push and pull strategy. So in fact, during my PhD studies, I actually did this one, because let's say 4 different species like Aegium and the Barbadans, Arborium. So that's why what we did is like, the Hilsut is the most commonly, most commonly used cultivar species, like, So that's why from the Barbadans, so Lakshmi cotton variety, we actually did that. We used it as a component in the IPM. So for my PhD studies, I had done that one. So the extract, leaf extract, because we
extract,
we had done, but not in this perspective. In which he asked. But that was only like, tritropic interaction context.
Yeah. So like Gangadhar, what we also here did, the repellents what we have used. So they are nothing but from the plants only. Yes. But we have not identified the chemical component in that.
Yes, sir.
Yeah. So we can use them.
You can directly use them.
We can use directly.
Not necessarily you don't have to go for the chemical analysis or extraction of it. Naturally, the food extract is more than enough. Our objective is only to either to push or to pull. That's all.
Yeah, definitely.
That's what I did. Like, I did not go for the infochemical, the cinnamon extraction. I didn't do. Directly food extract of the leaf from the one species. I had taken up the spray in the other species. That was one of the components involved in the IPMs.
Yeah, nice.
Sir, 2 more clarifications, sir. The one is regarding the pomegranate studies, push pull technology and pomegranate. That was an excellent study, sir. Actually, the first of its kind, the initiation of such kind of studies. But I'm just comparing. You proposed 3 models. Using of the attractant, the traps. Putting them in half of the probably the piece of the entire land, or putting in a periphery, and probably one is random distribution. If you are putting or installing those attractant traps in a periphery, the logic is that you can attract the moths very easily, which are entering into the field from the neighboring infested plant.
Yeah,
that's the logic or principle behind that. It seems. I'm just comparing the again back with the Kenya study.
Hmm.
the maize, desmodium, and napier.
Hmm.
In the case of napier and maize, and when you compare that model with maize, desmodium, and napier,
Hmm.
according to the result what you presented, sir, the combination of maize, desmodium, napier were the significant reduction of the stem borers in that case, the model as compared to the maize with only napier.
Hmm.
Taking the